Hi student, welcome to IGCSE Biology Intensive Seminar. Today I would like to share some tips and tricks on how to answer question in IGCSE Biology 0610 paper. For IGCSE gradings, the candidate can opt for core paper or extended. However, all the candidates have to take 3 papers. Grade A star, A, B, C, D, E, F, or G indicate the standard of a candidate achieved at Cambridge IGCSE, whereby A star will be the highest and G will be the lowest. U stand for ungraded, means the candidate performance did not meet the standard requirement for grade G. The first paper is paper 1 or 2. Paper 1 is for those students who opt for core paper, and paper 2 is for those students who opt for extended paper. For this paper, students are required to answer 40 multiple choice questions within 45 minutes. 40 marks will be contributed for this paper and it will be converted to 30% of the total paper. Question will be based on the extended subject content, including the core and supplement learning outcome. And this paper will be assessing from grade A star to G. This paper will be externally assessed. The second paper is paper 3 or 4. Paper 3 is for those students who opt for core paper, and paper 4 is for those students who opt for extended paper. Students are required to complete the question within 1 hour and 15 minutes. For this paper, students need to answer at least 6 to 8 short answer and structure questions. And 80 marks will be given for this paper, which will contribute 50% of the total paper. Same as paper 1 and 2, question will be based on the extended subject content and it will be externally assessed with the grades from A star to G. The third paper will be paper 6. Students who opt for core paper or extended paper will be taking the same paper. For this paper, students are required to complete the paper within 1 hour. There is 1 to 3 questions that will be based on the experimental skill in section 4. Total of 40 marks for this paper, which will contribute 20% of the total marks. Again, this paper will be also externally assessed with the grade from A star to G. Now, let's dive into one by one on how to answer certain questions. When you answer question, remember to keep your handwriting clear and legible. On the left side of the paper, there is a column marked for examiner use. Make sure you don't write anything in the left-hand side of the paper. Keep all your answer on the line on the question paper. If you wish to change your answer, remember to cross out your first answer by putting a line through it and rewrite. Remember, don't scribble over what you have written. Let's say if you have run out of space, you can use the white space on the another part of the exam paper for a continuation answer. Remember not to squeeze your answer by using a very small writing, which will make the examiners hard to read your answer. If you need to use a different space for a rewritten another answer or continue with your answer, remember to put a note to inform the examiner where is the continuation of your answer. Always remember you are taking biology IGCSE paper. So whenever you answer certain question, you need to always try to write as accurate as you can by using the correct biological terms. As I mentioned during class, don't use the words such as like it, they, effect, affect without further explanation. Because you always have to think about whether examiner can understand what you want to talk about. For paper 2 multiple choices question, always remember that there is no pattern in the letters order of correct answer. The same letter could be the correct answer for several answers in a row. So remember, do not let what you have chosen for the previous question influence what letter you choose. When you need to sketch a graph in the exam paper, remember to choose a simple scale which uses most of the grid. 1 marks will be given if you use most of the grid. Remember to state the x-axis and y-axis clearly together with the unit. Students, remember that x-axis is for the manipulated variables, whereby the y-axis is for the responding variables. Remember to plot the point exactly using pencil. Please do not use pen when you plot a graph. Try to join all the points with a line of best fit or a zigzag line. Remember that not all the curves have to pass through the point where the two axes meet. When the question requires you to write an equation, remember to read the question clearly, whether you are asked to give either a word equation or a symbol equation. Remember, do not combine symbol equation and word equation within the same answer. For example, on your left side, it is the equation for the anaerobic respiration. On the top, it is a wrong answer because it combines the word and symbol equation within the same equation. The bottom is the correct example of symbol equation or word equation. Now, let's learn how to answer the question precisely. Always remember to give a precise answer to the examiner so that they can award you the full marks. For example, state why magnesium ions are important for healthy plant development. A lot of students like to give general answer such as they are needed by the plant or they are needed by the leaf. Ask yourself why they are needed by the plant or why they are needed by the leaf. You need to use the theory that you have learned during biology lesson to further explain this question. Some students answered, magnesium is needed to make chlorophyll. Why magnesium is needed to make chlorophyll? So, the ideal answer will be, magnesium is a raw material from which chlorophyll is made. Example number 2. The fertilizer causes low oxygen and it affects animals in the water. So you need to ask yourself how and why they can affect the animals. So the ideal answer is the animal do not have enough of oxygen. Why? What happens if they do not have enough of oxygen? Remember that the animal need oxygen for their respiration. And how it will affect? If the animal do not have enough of oxygen, eventually they will die. Some students like to use a highlighted pen to highlight the keywords in the question. Unfortunately, highlighted pen is not allowed when you are taking IGCSE exam. So what you can do is using pen or pencil to underline or circle the keywords in the question. For example, please look at question 1. Name the tissue that transport the sugar made by photosynthesis to the other part of the plant. The first word name is important because this will tell you whether you should write a one-word answer or you need to further answer the question. So since here they stated as name, so you can give a one-word answer. So name the tissue, now talking about tissue instead of cell, that helps to transport. And the keyword is transport sugar that made by the important process known as photosynthesis to the other parts of the plant. So all the underlined words are the common words or the biological terms that we use during biology. So give yourself 3 seconds to think about what is the answer. Yes, the answer is phloem. Talking about common words, there are several common words which is usually can be found in the exam paper, such as like name, state, define, describe, and explain. If the question asks you to name or state, usually you just need to give one or few words of the keywords or the biological terms that used in biology. If the question asks you to define the term, so what you need to do is to write the definition for the term. The definition of the biological terms can be found in your textbook glossary part. When the question asks you to explain or describe, you know that you need to further explain on the biological term. As I mentioned before, when the question asks you to describe the graph, so what you need to do is you need to look at the pattern of the graph and explain the graph clearly. Marks will be given if you give a clear explanation. Remember not to use those vague words such as like it, they, which you might not know what do you mean. Furthermore, I need to emphasize on the spelling. Even though this is not a first language paper, however, spelling is also important. Wrong spelling will not be given marks. Structured identification question also can be found in the exam paper, especially paper 3 or 4. So, students need to know certain structures of a plant or human body clearly, such as like the structure of reproduction organ, the structure of a heart, as well as the structure of a flower. Some question might ask you to label or identify the part of the organs, such as like this question 4. Use the label lines to identify the flowing on the drawings of a flower, petal, sepal, and stamen. 3 marks will be given for this question. So, student, you need to know the structure of a flower clearly. And you need to find the structures on a diagram of a flower that you may never have studied. Draw a label line clearly to the structure and write the name next to the labeling line. However, line is not given, but you need to write besides the labeling line. So be careful not to miss this question as line is not given. Remember, even though it's a biology paper, you still need to prepare calculator with you when you enter the exam hall. When there is a question require you to calculate the numbers, okay, you may need to find the figure from a table or from the graph. When 2 marks is given, remember to write all the working steps for your calculation, because the working step also will give you points. As I mentioned during class, always remember to show the units in the calculation, because in the marking scheme they mention that units is needed. If the units are not given on the answer line, then you must make sure that you need to write them after your numerical answer. No units, no points. Sometimes you will be asked to carry out a calculation and add the result to the table. Always remember to express your answer in the same way as the figure given in the table. For example, in the table, the numbers is given with 2 decimal points. So even though your answer is a whole number such as like 16, but you need to write the 2 decimal point like 16.00. If you use a calculator, remember to round up or round down the figure. Don't copy all the figure after the decimal point. Some question might require you to give the answer in whole number or 1 or 2 decimal point. Remember to read the question clearly and give the answer that they want. During your Mock 1 exam, some of you calculate and give the correct answer. Why points is not given? It is because the question requires you to give the answer in whole number. However, some of the students write all the figure that's shown in the calculator. Please remember. And some of the students didn't give units after their answer, which also you will get 0 for your answer. So remember, don't make this kind of careless mistake during your exam paper. When they ask you to draw a table in the question, so always remember, use a ruler and pencil to draw the table. Do not use pen or freehand to draw the line, because make sure you need to rule the lines for the columns and the row. 1 mark will be given if we are using ruler and pencil to outline the table. Remember to write the heading for each column and row of the table. Inside the heading, you need to write down the units. For example, like volume of water, bracket, cm cube or ml. Time, bracket, minutes. The unit should be written after the oblique line. And remember, since you already mentioned the units at the heading, please do not put the units in the table space when you write down the number. Make sure you are using the same number of decimal places in each column or row. Means, for example, some of the question, it can be 16, it can be 16.5, it can be 16.25. So 16.25 contain 2 decimal places. So all your answer or all the numbers need to be 2 decimal space. So what you need to write? 16.00, 16.50 and 16.25. So all the number need 2 decimal place. If you ask to complete a tally chart for a recording data, what you need to remember again, you need to use ruler to draw the table and give a clear heading. How to do a tally chart? You need to record the number by using stroke and give an oblique line to represent 5. Remember to include another column to show the total numbers of the stroke. For table drawing question, usually 3 to 4 marks will be given. 1 mark is given if you draw the table by using pencil and ruler. 1 marks will be given if you are giving the correct heading together with the units. 1 or 2 marks will be given if you write down the numbers in the table space clearly and correctly. When the question requires you to construct a line graph, remember to use a cross or a dot in a circle for your plot points. Students are not encouraged to use a single dot when they plot the point. Why? It is because a single dot may not be seen easily or clearly by the examiner when you have drawn your line. When the question requires you to plot 2 lines on the graph, remember to use 2 different symbols to represent 2 lines of graph. And you must label each line carefully and clearly, or you can use a key. Remember, when you draw line graph, you need to use pencil. So, please use a pencil for both lines. Please do not use pen, whether blue or black pen or even highlighter pen when you construct a line graph, because it might not be able to show up on this scan script. Remember to read the question carefully before you put a line in the graph. Some questions may require you to give a straight line of best fit. Some may ask you to give a smooth curve of best fit, or some just need you to join the points by straight lines. Always remember to prepare a sharp pencil so that the graph that you draw will be thin and smooth. Remember that the line of best fit doesn't have to pass through all the points where the two axes meet. And do not extend your line graph beyond the last plot point. Not all the lines need to start from 0. And student, you must remember, x-axis is for manipulated variables and y-axis will be for the resulting variables. If you draw the graph upside down, you will not get any marks for that. Remember. Besides that, when you label the axis, must always include the units. And another thing that all of you need to be aware is that sometimes they will ask you to draw a line graph based on a table. But for example, some of the question, the table might give you the numbers of bubbles produced. But when they ask you to construct a line graph, they might change the story to, they ask you to give the rate of photosynthesis or rate of respiration on this. So don't make the mistake of labeling the graph wrongly. So if the line graph asks you to give the rate of photosynthesis, please give the rate of photosynthesis instead of numbers of bubble. And remember, please do not squeeze your line graph. Please draw at least more than 50 or 70% of the grid papers given. As I mentioned just now, 1 marks will be given if you draw the graph as big as you can. But of course, don't exceed or extend the line graph. Now, I would like to teach all of you how to differentiate between bar chart and histogram. Most of the time, IGCSE biology paper required you to draw the bar chart, but I can see some of you are drawing histogram in the exam paper. So, what is the difference between bar chart and histogram? For your information, bar chart have separate columns that they do not touch. There is a gap in between each bar. For histogram, they have columns, but they will touch each other. Bar chart is usually used to show data on the discontinuous variables, for example, blood group, eye color, and etc. For histogram are usually used to show the data on the continuous variable, such as like length, mass, speed, and volume. However, you need to read the question carefully because they will state clearly whether you should draw a bar chart or a histogram for the question. Now I would like to show you the example of bar graph and histogram. So this is an example of a bar graph. As you can see, there is a gap between each bar, and the gap between each bar need to be equals. As I mentioned before, remember the x-axis will be the manipulated variables and the y-axis will be the responding variables. However, in bar graph, if let's say they are giving you two different data to plot, you cannot shade the graph or color the graph. So what you can do in order to show the examiner or allow the examiner to differentiate between two different data, you need to label the data clearly at the axis. And always remember to write the title on top of the bar graph. This is the example of histogram. As you can see, there is no gap in between. When you draw a bar chart, remember to use a scale which uses most of the grid provided on the exam paper. Repeat again, please do not squeeze or make your chart become too small, as mark will be given if you use most of the grid. And remember to draw the chart by using pencil. Remember to rule the column evenly so that they are having the same width. Please do not give a column with different sizes. Space between the column on the x-axis should be the same. And your y-axis should be properly scaled with equal interval and must be labeled clearly with the units. The lines or blocks can be arranged in any order based on the answer given in table. But in order to make comparison, it helps if they are arranged in descending or ascending orders of size. Remember, as I mentioned previously, you must identify each block by putting a label directly underneath each block at the x-axis. Please again, do not shade the blocks or color code them, as it couldn't be seen in the scan script. If you shade the blocks or the columns, no marks will be given for your bar chart. When you draw a histogram, you need to choose a scale which uses most of the grid provided. And remember to draw the histogram by using pencil instead of pen. Same as bar chart, the x-axis represents the independent or manipulated variable and is continuous. Remember to properly scale and label with appropriate units. For histogram, the block should be drawn touching with no gap in between. The area of each block is proportional to the size of the class. It is usual to have a similar size classes, so the width of the blocks are the same. The blocks should be labeled by putting the classes ranges underneath each blocks or by putting the lowest number in each range under the left-hand side of the relevant blocks, whereby the y-axis will represent the number of frequency and should be properly scaled with equal intervals. And remember to label with appropriate units. However, bar chart is frequently can be seen in the exam paper compared to histogram. So student, please be careful and read the question properly whether they request you to draw a bar chart or a histogram. Sometimes the question might require you to draw a horizontal or vertical bar chart. Please read the question carefully. Planning and investigation questions frequently can be seen in paper 6, alternative to practical. When you read through an investigation, try to find out three main things. What is being changed, which is known as independent or manipulated variables. What is being measured, which is the dependent or responding variable. What is being kept the same all the time during the experiment, which is known as control variables. For example, look at the example number 10. In a question, to investigate the effect of temperature on the enzyme activity. So, the independent variables or the manipulated variables will be the temperature. And the dependent variables or the responding variables will be the enzyme activity. So, what is the control variables for this example? So, since the temperature is something that you need to be changed and enzyme activity is something that you need to observe, so something that need to keep all the same will be the volume of the enzyme, the concentration of the enzyme, okay, the pH of the enzyme. And I remind all of you again, when we talk about something related to liquid, remember to use the words volume instead of amount. And another thing is that they might ask you how to maintain the temperature for the experiment. So how we shall maintain the temperature of the experiment, we need to use the thermostatically controlled water bath. When the question asks you to plan an investigation, remember to be specific and precise. Don't use the words such as like some, several, few, and same. You need to mention the numbers clearly. And also for manipulated or independent variables, please write at least 3 different options for the variables, such as like for this question, they ask you to investigate to the effect of temperature. So you need to give me at least 3 temperature instead of 2. Maybe you can give me example like 10 degrees Celsius, 25 degrees Celsius, and 37 degrees Celsius. Don't just give me 2 because sometimes the marking scheme require you to give at least 3 manipulated variables option. And also, when you need to give me the dependent or responding variables, you need to explain clearly how you can obtain the result. Example like the bubbles produced. So how you can obtain or how you can calculate the numbers of bubbles being produced. Use a timer. How long you need to calculate the bubbles? 5 minutes, 10 minutes. You need to state clearly. Some investigation need to have 2 parts, which are experimental and control. Experimental measured the process being studied and contains the living organism, part of an organism, or enzyme being tested. For control, it is exactly same as the experimental, except that the living organism will be missing or replaced by something non-living. The controls show that the results are due to the activity of the living organism and is not due to the apparatus or an environmental factor. For example, when we investigate the effect of enzyme on the rate of reaction, for experimental, it will be added with some enzyme. But for control, water will be used to replace the enzyme so that it can show that the result are due to the activity of the enzyme, but it's not due to the apparatus or any other factor that might affect the rate of reaction. Always remember to mention that the experiment or the investigation should be repeated at least 3 times to increase the reliability of the result. If the same result or similar result are achieved all the time, then this experiment or this investigation are reliable. The precision of result taking may not be very good. For example, if you are measuring using a syringe or measuring cylinder, it may be difficult to measure down to the nearest cm cube. Always remember to give the quantity in appropriate terms. As I mentioned earlier, avoid the use of term amount. Remember to be always precise and use the correct terms. When the question requires you to record an observation, remember to write down exactly what you can see based on the photograph or diagram on the paper. Some of the students tend to describe the color of the plant or color of the animals, whereby the photograph or the diagram doesn't show the color of the animals or plant. Remember to describe the difference of similarity clearly based on the photograph or diagram. Measurement may need to be made and magnification calculation also be required. Look carefully at the pictograph. Sometimes magnification is given as some of the diagram will be enlarged, such as like times 100. If you are asked to calculate a magnification, always remember the formula of magnification in your brain. So, when you calculate a magnification, you need to follow these steps. Remember to measure the structure in the photograph in millimeter, not cm. Look for the actual size of the object. Sometimes you will be given and sometimes they will ask you to measure the diagram. Remember to round up or round down the answer from your calculator. Usually magnification are given as whole number. So please do not give the answer to one or more decimal places. Actual sizes could be given as whole number, or you could include all one or two decimal places. Last but not least, my students, remember to smile while doing your exam paper. Remember to breathe, inhale and exhale, because your brain needs energy through respiration. My Year 11 students, good luck and all the best for your IGCSE. Thank you.